Hi, my name is Maya Wright. I'm a sales application specialist at Fluidic Analytics. And today I'd like to give an introduction to a novel method for measuring protein interactions in solution called microfluidic diffusional sizing. So how does microfluidic diffusional sizing work? So what happens is that you load a labeled protein sample onto a microfluidic chip. This is a schematic of the chip. The protein sample gets injected into the microfluidic channels and meets with a stream of water. And because of the micron scale lengths of the chip, the only way that the protein mixes with the water stream is by diffusion. So you can imagine that smaller proteins will diffuse more quickly and larger ones more slowly in this channel in the same amount of time. And by measuring the amount of diffusion that happens, we're able to calculate the hydrodynamic radius of the proteins. To do this, we split the streams at the end into two, into the diffused and undiffused portions, and the ratio of the fluorescence signal of the diffused and undiffused gives us a size, and the sum gives us a total abundance as well. So in order to measure interactions, you would repeat the same analysis, but pre-mix the labeled protein sample with a binding partner that's unlabeled. And by repeating the same analysis, we can see whether the diffusion is affected by binding. If there's a binding interaction, the protein diffusion will be slower, and you will end up with a larger hydrodynamic radius overall. So by measuring changes in protein size and their complexes, we're able to measure protein interactions and to determine their KDs of interaction. So the product that does this is called the Fluidity One-W. To give a few specs, the sizing range is from 0.7 to 20 nanometers in radius, which covers most proteins and their complexes. It's currently available at two different wavelengths, the 488 nanometer wavelength and 647 nanometer wavelength. The sensitivity of both systems is about 1 nanomolar of ELISA dye, with KD ranges down to 100 picomolar at the lower end. The sample volume requirement is very small, with only 5 microliters required per radius measurement and about a 9-minute readout per data point. The different application areas include looking at protein interactions, but anything really that causes a change in size. So protein-lipid interactions, interactions with DNA, small molecule protein interactions, peptide protein interactions, and so on. And crucially, because we are based on detecting by fluorescence, we can measure protein sizes in crude and complex backgrounds, including cell lysates, serum, and cell culture medium. So to summarize the different spaces we are able to address, we can look at protein interactions in backgrounds such as blood plasma, do quality analysis of proteins during bioprocessing, and also to characterize unknown protein binding partners based on the hydrodynamic radius that we measure, we can gain insights into the molecular weight of the binding partners. So to benchmark the Fluidity One-W technique against techniques that are currently on the market, we compared the Fluidity One-W data with microscale thermophoresis and surface plasmon resonance. Here we looked at the interaction of a therapeutic antibody nivolumab with the extracellular domain of PD-1 protein. So what we did was to label the PD-1 protein with a fluorophore to keep its concentration constant and to vary the concentration of the unlabeled binding partner nivolumab. We could see here in blue with the Fluidity One-W that as you increase the binding partner concentration, you see an increase in the hydrodynamic radius, showing that there's a binding interaction. We compared the KD that we obtained to that of MST and SPR measurements, and you can see on the right here that the KDs of interaction are in the same range as MST and SPR, but with additional advantages including in-solution measurements without the need for surface immobilization. and also the ability to tell stoichiometry of binding as well based on the hydrodynamic radius. So we also looked at the interaction of two proteins in two different buffer backgrounds, in PBS and in cell expression medium. Here we were looking at the interaction of protein A with IgG. As with the previous study, we kept the concentration of the labeled protein A constant and varied the concentration of the unlabeled binding partner IgG. We could see that the radius increases with increasing binding partner concentration, showing that there's an interaction. And we were able to fit the curves for the two different buffer backgrounds to measure the KDs of interaction. The KDs obtained were in the similar range for both backgrounds, showing that the protein A IgG interaction is unaffected by the matrix. Based on the final size measured, which was 11.5 nanometers, we could also tell the stoichiometry of binding. The hydrodynamic radius measured is too large to correspond to a one-to-one binding of protein A and IgG. And by comparing to simulated structures of protein A binding to IgG, we were able to infer the stoichiometry of binding to be about 3-to-1 protein IgGs per protein A. Finally, we tested the interaction of the SARS-CoV-2 antibody against the receptor binding domain of SARS-CoV-2 in pure buffer and in human serum. Using the 647 nanometer detection, we were able to see that the KDs of interaction in the buffer compared to in the human serum background, that the interaction KD is roughly the same for SARS-CoV-2 anti-spike protein and the RBD of SARS-CoV-2. So in summary, we've developed a novel in-solution approach for protein sizing and interaction analysis, both in pure buffers and in complex backgrounds such as cell culture medium and serum samples. Protein samples can be analyzed in about 9 minutes with a very low sample volume requirement of only 5 microliters per run, with additional insights gained into interaction stoichiometry, and as well as the conformation of your protein. Thank you very much for your attention.